Hello, we're glad you've joined us for this live webinar, Flow Cytometric Approach to Probiotic Cell Counting and Analysis. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Beckman Coulter Life Sciences. Beckman Coulter Life Sciences develops, manufactures, and markets products that simplify, automate, and innovate complex pharmaceutical quality control testing. More than 275,000 Beckman Coulter systems operate in both diagnostics and life sciences laboratories on 7 continents. For over 75 years, our products have been making a difference in people's lives by improving compliance and reducing cost of ownership. To learn more about Beckman Coulter Life Sciences, please visit www.beckman.com. So let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. I now present today's speaker, Dharlene Tundo, a quality control senior microbiologist at VitaQuest International. Dharlene earned a Bachelor of Science in molecular biology from Montclair State University, and she has amassed more than 10 years of diverse experience in research and in the pharmaceutical industry. She has contributed to the successful launch of 2 oncology drugs, Cyramza and Portrazza, at Lilly Pharmaceutical. Dharlene's research interest focuses on the study of Lactobacillus and Bifidobacterium species. She is dedicated to developing a method to analyze and obtain cell count differentiation on viable and non-viable bacterial cells for the manufacture of probiotic supplements using the Flow Cytometric approach. Dharlene Tundo will now begin her presentation.
Thank you very much. And like I said, today we are going to be talking about the flow cytometric approach to kind of optimize the traditional method, which is plate count, mostly used for all microbiologists when it comes to determine the viability of probiotic cells. And the reason why we decided to look into this new approach, it was with a purpose to kind of optimize the timing, because this is a great benefit for the industry, especially when production of probiotic supplements or other products are in high demand for a large-scale production. And we see that in many, many ways. When we go to the market, we see from supplements to facial masks to drinks to so many products out there that definitely optimizing or coming up with a new method that saves time to the manufacturer is of a great benefit, and definitely the turnaround time would be very profitable. So let's talk about the market growth. Due to the market growth, there is a lot of different products that are being manufactured at large scale. Therefore, there is a high need for these products to be available to the consumers. Usually, when we do the viability determination for these products or the raw materials, we have to plate these samples, and then it takes about 3 days. So we're talking about a little time-consuming method, and that doesn't allow the manufacturer or the vendor to release the products on a timely manner as they desire. So just to tell you a little bit about the traditional method, it's the plate count, and it usually requires multiple dilution of the product, because usually the product comes in powder form or freeze dry. form, and therefore it requires different dilutions. Some products might require less dilution or more dilutions than the others. Usually takes 3 days. In those 3 days, you are able to collect the results, and that will let you report the counts. So the product is ready to be manufactured at that point. So with this method, what we do is we make the dilutions, and then we will plate them in MRS agar, or then man-rugosa and sharp agar, and then we would incubate it in these chambers that are anaerobic. It contains a gas pack that it would maintain the chamber oxygen free. So this organism, either Lactobacillus or Bifidobacterium, will grow at its optimum environment. Then we will incubate for 2 to 3 days at 37 degrees, and then we will be ready to collect those results. So the method consists on plating these products in duplicates. For each single sample that we test, we would have to duplicate the sample. So for each sample, 2 plates. Then at the end, what you see is all these colonies on single plates, and we would have to count each of these colonies and report in CFU per gram, or colony forming units per gram. And as you can see, these plates are really, really covered in colonies, and sometimes it's really, really tricky, and it can be challenging as well to count all these colonies and have to report a result in a timely manner when you have, let's say, 20 or 30 samples in one day. As you can see, this is really, really time consuming. So usually the 2 different type of species that we deal with in the manufacture of these supplements are Bifidobacterium and Lactobacillus. And just to give you a little overview about these 2 species, they are really challenging at times, because what happens when we incubate these samples, if we have single colonies or single species on a plate, the Lactobacillus usually can be differentiated from the Bifidobacterium. And we do that by eyesight. For example, Lactobacillus colonies will be a little bigger, and Bifidobacterium will be a little smaller. So sometimes we will have to leave the Bifidobacterium plates for an extra day. This way we can see the colonies and have a more accurate count. And this is one challenging, because when we have a blend, let's say a raw material sample that contains Bifidobacterium and Lactobacillus, we would have to treat this sample differently than our average sample. And because of that reason, it's a little trickier to kind of differentiate between the 2 of them. And also we will have to, in some cases, duplicate it in 4 plates, where you can kind of modify the incubation process for each of them. This way you would obtain a more accurate result. So this plating method, it's not that accurate when it comes to determining the metabolic activity and also physical characteristics of these different species. So why do we look into flow cytometry? Well, flow cytometry, it's a lot more rapid method compared to the traditional. And it has the benefit that provide us more data than the regular plating, which allows us to see if how many live cells or viable cells we have within a sample, and how many dead or non-viable cells we have. And this is a great thing, because with the plate, we don't know how many dormant cells, or we have a certain number of non-viable cells, because the the culture dependent just cannot provide us with a number. And another thing is that it's cost effective. We are able to run samples and get the results the same day. And that allows us to be more productive because we are able to release those raw materials in a more timely manner. And then manufacturing can go ahead and produce all these products that need to be released to the market. So it is time efficient. It saves us 3 days compared to the traditional method. And also the beauty of this instrument is that not only we're able to do single sample for using the tube loader, but we also have the plate loader, which has a 96 well plate, and you can run as many samples as you want. It's great. It's easy to use. You have as many samples as you want. You load your sample, your stain samples, and it has a feature for auto record, and you can basically load your samples and walk away, and the instrument will do everything for you. So that's a great benefit. This is just a little overview of comparison compared to the plate count versus the flow cytometry method. And this shows you in detail kind of like where we start when we receive the sample in the quality control department, and then the multiple dilutions we have to do in order for us to plate versus the serial dilutions for flow cytometry, which are less. And in the plate count, sometimes we have to dilute the sample up to 10-fold dilutions, to 10 to the 10th. With flow cytometry, I usually do 10 to the 5th for raw materials. And if we are analyzing any other type of form like solid dose, we would do 10 to the 4th, which I found very, very optimal for collecting this data, and it gives you beautiful graphs and very minimum electronic noise. So compared to the traditional method, you can see from the time we collect the samples and we make the dilutions, and then we stain our cells and we incubate them, it only takes 2 hours. And that is like a very large timing, 2 hours, and it could be, in those 2 hours could be 10 to 20 samples, depending on the prep time and depending on the technician, how quick we can get those samples ready for the flow. And the big question is, why did we pick Cytoflex? Well, Cytoflex, it was some instrument that we thought it could be the best thing or best option for us. Number one, it was because it's super modern, automated, doesn't take much room, and we literally have it in a bench top, like in a small table in the lab, and it's a great instrument. It doesn't require too much space, and the beauty and the main thing of this instrument is that it was able to deliver to us what we were looking for. And what we're looking for is a flow cytometer that is able to read or detect cells that are small as 0.5 microns, which are like the size of the cells that we are dealing with. This flow cytometer is able to detect cells as small as 200 nanometers, which is really, really sensitive and high tech. And we really fell in love with it, and we were ready to give this instrument a try, and we were not disappointed at all. The results that we obtained with this instrument were in accordance to the results that we had with the plating method. And that's when we really agreed that this was the right instrument for us and that we were on the right track. Just to give you a little overview about the model that we are using, we have the Cytoflex flow cytometer blue, red, and violet series. That means that it has 3 different colors. the red laser, the blue laser, and the violet. And basically, from the blue laser, we get 5 channels from the 488 nanometer laser. And we get 3 channels from the 638 nanometer laser, and 5 channels from the 405 violet laser. And basically, we can get up to 13 fluorescent detections with this instrument. And right now, because we are using Cyto 9 and PI, we found that these having these instruments with only red and blue lasers activated, it's more than enough. But we can always go back and get the violet laser active if we ever need it. But for now, it's working perfect. It gives us great results with these 2 lasers, and we haven't had any problems with it. So this is just a top view, and you can see where the lasers are located. They're like color coded, and then you can see to your right the picture of the front, and right where the arrow is, this is where the plate loader goes. And this instrument also gives you the benefit that if you didn't purchase this instrument with a plate loader at the beginning, you can always upgrade it, which gives you a lot of flexibility depending on the high demand of samples that you need to run. So just a few tips for success that helped me a lot during the investigation and looking into different sample data that we obtained at the beginning. We figured that having the optimal dilution sample, it was really critical. Sometimes if we had multiple dilutions, let's say, for example, 10 to the 8th or 10 to the 9th, which is the dilution that we use for most of these products when we do traditional plating, we realized that we were not picking up those events in the flow cytometer. Therefore, the sample was too diluted, and even though we stain it properly and we follow the procedure, we figured that it wasn't the optimal dilution needed for this analysis. And after different trials, we found that 10 to the 4th and 10 to the 5th were the best dilution for this type of products. So we learned that, and we also learned that threshold and gain settings were super, super important because these cells are super small, and at the beginning we had a hard time figuring out where or what was the best threshold for these type of cells, and also to not confuse the cell population with any other noise found or detected by the instrument. So we really look into threshold settings and different levels, and until we figured out that what was the best. And I'm gonna show you later on a comparison between 2 different settings, so you will understand better what I'm trying to explain to you. Another thing is that these fluorochromes are light sensitive, and it's very important that you protect those little vials that contain the dyes, protect it from the light. Me personally, I don't like to leave them out in the room temperature too long, so try to prep your samples in a timely manner and stain them and incubate them, and then put your dyes away, and also protect your samples from light. You have to make sure you protect your samples from any contaminants, because this instrument is super, super sensitive, and it's going to pick up anything that you load in that sample tube. So basically the instrument will do whatever you tell the instrument to do. And therefore, having great aseptic techniques really, really help and takes you a long way. Then also we have to ensure the flow rate is calibrated. For this instrument, we did a calibration for the tube and also a calibration for the plate loader, just to make sure that we were having accurate results. And we were not missing on any settings when we first purchased this instrument. So the experiment that we designed was for of different species, but the ones that I'm going to talk more about, it's within the Lactose and Bifidum. And just because Lactobacillus was our first species to start with, Lactobacillus is very robust, so it's very easy species to play, to stain for flow cytometry compared to Bifidum species. And the membrane integrity was assessed using Cyto 9, which stains everything in your sample. So anything that has DNA, Cyto 9, it's going to bind to it, either if it's viable or non-viable. And then as a counter stain, we use propydium iodide, or PI, like we know them. And what this does is it's going to bind into any dead cells or any damaged cells, and it allows us to have a differential counts for viability and non-viability. These are the settings that we used, and I find the gain settings set to 1,000 for 4 scatter and 5 scatter provided me great results and good resolution. And also the threshold for the Lactobacillus, I found best set at manual, and also 50,000 for 4 scatter and 10,000 for the 5 scatter. And also these settings were set to logarithmic because it's a better representation of the raw data that we collected, and it definitely shows you a better presentation of the histograms and also of the 2 populations of dead and live cells. So this is the first sample, and this is a control sample that we loaded, and this one is Lactofermentum, and this is on stain, and this is what we saw first, and you can see the lactose into the to the left, and also in panel B we have LGG, which is Lactobacillus Rhamnosus. LGG is very, very popular within the products that we process, so it's very, very utilized in many products. And then here you see the difference when we stain the sample. We have on the first panel the bacterial population detected on the previous slides that I just showed you, and the histogram on the middle shows you the beautiful peaks representing the live and dead cells. And these counts were at the beginning, this is just preliminary data when we started running the samples, but we definitely were able to find the 2 populations and also obtain this nice graph with a histogram, and that allows or confirmed for us to see that this was the bacterial population we were looking for. This is just another example of Lactobacillus Rhamnosus. These in particular I find very easy to work with the Rhamnosus. We did the same procedure, staining using Cyto 9 and PI, and the threshold and gain settings are exactly the same as I show you at the beginning. And the first panel you can see the bacterial population, and the second one shows you the histogram, and the third panel shows you the dead cells versus live. And we were looking into expected count of 350 billion per gram, and we got 423. And it's not a surprise, because when you look into the plating, you won't get as many cells as we got into the with this flow cytometric method. And this is because in the plating, you're not able to see the dormant cells. And the dormant cells are able to grow. They're not able to show the colonies, but they are not dead. That means that they are metabolically active. And the flow cytometer is able to give us those cell counts, which we cannot get with a plate. And this is one of the our favorite things when we get these numbers, because it shows more accuracy comparing to the traditional method. This is Lactobacillus Acidophilus, and you can see the little difference between the previous one and this one. It's still a lactobacillus, but it's a little cleaner, and the data didn't present as much noise as the other ones. And this is something expected because depending on the raw material sample that we are prepping, there is a cryogenic or cryoprotectant covering these cells. And this is just part of the freeze-dry method that these cells go through before we get them into our lab or into our company in powder form. And therefore, some of them might have a cryoprotectant that is thicker layer than others, and therefore it can be a little more challenging for sample preparation or cell staining process. And some of those samples are going to present cleaner data than others, but because of all the controls that we ran at the beginning, we have confirmed which are our bacterial population. And we also, each sample that you are seeing here, we also plated, which we have the data to support our experiment and confirm that we were on the right track. So for example, on this one, we have 15 billion per gram, and we were expecting 13 billion per gram. Usually in a plate count, we will obtain something around 13 billion. Sometimes we will get a little over, but not as much. And so these results were great, and were in agreement with the plate results that we got. This is a comparison between two different settings. And this is why I said it's so important to understand and to know what is the optimal settings for your threshold and for your gain settings when it comes to obtain a good data for these samples. And this is a great example showing the bifidolongum. And to the left, you're seeing a threshold set for 50 and 10,000 for 4 scatter and 6 scatter, respectively. And you can see how you have these two populations separated, and you can gauge them easily compared to the one to the right, where we have a setting of 5,000 for 4 scatter and 6 scatter, respectively. And it's a little more challenging, and you can see the difference in the graph. So definitely it makes a big, big difference, and it's super, super important that we have the right settings for each of these samples that we deal with. And some of the challenges that I found included the sample prep, like I mentioned. Therefore, I had to optimize the sample prep. Some of the samples, when I found that the cryoprotectant was too thick, I had to put the sample in water bath. And this is one example with the bifidolongum. I had to make the dilution, and then I had to put it in the water bath set at 45 degrees for 15 to 20 minutes. And then I went ahead and completed my dilution if I needed, and then stained the sample. After several trials, I figured that doing this treatment for the sample prep provided me better results than just going straight out and make the dilutions and stain the samples, because first of all, sometimes it presented too much particulates other than the cells, and also require a lot of fixation with formaldehyde. The data is not shown here, but those were the things of trials that were performed in the lab to get to this point and have to, were able to provide this cleaner data or the separation of two populations. This is a clear example of the settings again. So this one is for Lactobacillus Rhamnosus. On the left side, you see in the bacteria population, when the settings were set at 50,000 for 4 scatter and 10,000 for size scatter, versus 5,000 for scatter and size scatter, you can see the amount of noise outside the gate. And this also was another part of our studies in seeing how the settings, when they are properly set, can make a big difference and can also provide a cleaner data and more accurate numbers. So in conclusion, after doing several trials, we figured that we would do comparative studies, because we want to make sure that we really got accurate data, and that the method of flow cytometry was really going to increase our quality control systems. We had to do a lot of platings, and this was a big study for us, where we had to plate each sample that we analyzed in the flow cytometer, and we had to compare and count and put a whole report together. And what we found was that this flow cytometry approach is really applicable, and it's reliable, and it provided what we were looking for. And we are really satisfied with the results. Even though this is all preliminary data, it's great advancement in traditional microbiology. And we also confirm that we are in the right track, because the results that we got with the flow cytometer were in agreement with the plating count. And not only that, the workflow improved big time. We are able, with this new method, to release products in a timely manner. We don't have to wait for 3 days to release the raw materials for our production crew start to making the supplements. And it makes a big difference, because in our company, we do process or test using the traditional method about 2,500 samples per year, and that is a lot. And having 10 to 20 samples per day and plate them and have to sit down and count these colonies, I mean, sometimes you don't have time for that, right? And this method of flow cytometry, it's pretty sweet. It's very straightforward. It's so easy to use. The instrument is super modern, and we are really happy with it. So the improvement on the timing and the workflow definitely is a plus. And definitely this will be something that moving forward will replace the traditional method. And we will continue on investigating other type of species and other type of raw materials for probiotic supplement manufacturing. Right now, we are analyzing 20 different species within the Lactobacillus and Bifidobacterium. So it's a lot of data that we are looking into, but it's promising a lot. And this definitely would be a big innovation to the manufacturing of probiotics in large scale. And this would definitely move traditional microbiology to the next level. So if you have any questions, feel free to let us know. And I also have provided my information at the end. And feel free to contact me with any questions that you have or any other things that I might not cover in these slides that you are interested in knowing, and I'll be more than happy to answer those questions. Thank you very much for your time. It's been a pleasure, and I hope this was very informational and educational for everybody.
Thank you, Dharlene, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Dharlene will answer as many questions as time permits. And the first question for you is, how reliable is the flow cytometric technique in comparison to the traditional plate count method and its main advantage?
It's very reliable because of all the data that we have collected. Using the traditional method, which is our platform that we have been using for years and years, and we have confirmed and verified over and over the results that we obtain compared to the plate and also with the flow cytometer, and it has been in good agreement, which tells us that it is reliable and that it is a great approach to use, and definitely a technique that we can trust and that we can rely on.
Great. Thank you very much for that one. Your next question is, what controls were used during this experiment?
Thank you for your question. For the controls, we use unstained samples, and unstained samples for each different probiotic cell type, and for each different type of samples, either was raw material or chewable form, we use non-stain. And we also did fixation of samples using formaldehyde to confirm that we were seeing or detecting the bacterial populations that we were looking for.
Okay, that is great. It looks like we may have time for one more question today, and that will be, was compensation needed for samples stained with Cyto9 and PI?
Not at all, because the spillover of these samples, it's very minimum, and therefore there was no need to set up for compensation between the two lasers using these two fluorochromes. So it wasn't necessary, which was of great advantage because the data collected using Cyto9 and PI was straightforward.
Okay. Well, that is great. I would like to once again thank Dharlene Tundo for her presentation. Do you have any final comments for us today?
Everybody can reach out to me with questions that we covered today in this section, and also any other questions that we didn't cover here. I will be more than happy to answer. It's a lot of data, a lot of information to share, and sometimes even if we want to cover everything, we don't have time, but I'll be more than happy to answer more questions referring to the experiment or any other questions related to the instrument.
Okay, very well. Well, thank you again. And I'd like to thank the audience for joining us today and for their interesting questions. Questions we didn't answer today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. This webcast can be viewed on demand through August 2018. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.